Please subscribe, like, and share. It really helps us out. And of course, if you have any questions, comment below and we will answer you as soon as we can. Welcome to our video series on IGCSE Co-ordinated Sciences. This is Biology Unit 4.1. In today's lesson, we will be learning about nutrients. If you haven't seen our previous videos, click on the card above.
Firstly, let's look at food groups. To start with, you need to know the definition of nutrition. This is the taking in of nutrients. These are organic substances and mineral ions, containing raw materials or energy for growth and tissue repair, absorbing and assimilating them. You need to know the 7 food groups, their importance to an organism and their sources. Firstly, carbohydrates. These are starch and sugar. Starch is used for the slow release of energy. It is found in pasta, rice, bread, and potatoes. Sugar is used for the quick release of energy. It is found in candy, fruits, and soft drinks. This table can be downloaded from the link in the description. Next, protein. Protein is used for growth, repair, and it can be used for energy. It is found in meat, fish, tofu, nuts, and eggs. Now, fats. Fat is used for a store of energy, heat insulation, organ protection, acting as a shock absorber, and to build cell membranes. It is found in butter, cream, cheese, oil, avocado, meat, and fish. Moving on, vitamins. We will only look at 2 vitamins here. Firstly, vitamin C. Vitamin C is important because it promotes strong blood vessels and skin. It is found in citrus fruits, average fruits. Secondly, vitamin D. Vitamin D is important because it allows calcium to be used. It can be found in egg yolk, oily fish, for instance, salmon, and sunlight. Next, minerals. Again, we are just going to cover 2. Firstly, calcium. This is important because it makes bones and teeth. It can be found in dark green vegetables, milk, and bones. Secondly, iron. Iron is important because of hemoglobin production. It can be found in red meat, liver, egg yolk, and dark green vegetables. The next food group we will look at is fiber. Fiber is important for bulk up feces, prevent constipation, and prevent bowel cancer. It can be found in oats, bran, pulses, vegetables, and fruits. Lastly, water. Water is important for hydration, chemical reactions, and body fluids. It can be found in water, of course, and fruits.
Now let's look at deficiency diseases. You need to know the deficiency symptoms for vitamin C, vitamin D, calcium, and iron. Firstly, vitamin C deficiency. Vitamin C strengthens the blood vessels and skin, so vitamin C deficiency will cause overall body weakness, swollen gums in the mouth, nosebleeds, and in extreme cases, it will cause a disease called scurvy. Scurvy causes gum and bone pain and can lead to teeth falling out of gums. Next, vitamin D deficiency. Vitamin D allows the use of calcium, which strengthens bones and teeth, so vitamin D deficiency will cause weakening of bones and teeth, and in extreme cases, it will cause a disease called rickets. Rickets is a disease caused by the weakening of the bones, and the body and legs get bent because the weight of the body is too much for the weak bones. Now, calcium deficiency. Calcium strengthens bones and teeth, so calcium deficiency will cause weakening of bones and teeth, rickets, and in extreme cases, it can cause a disease called osteomalacia or osteoporosis. Osteomalacia is the softening of the bone. Osteoporosis is the fracture of the bone due to softening of the bone. Lastly, iron deficiency. Iron is used to make hemoglobin, which carries oxygen for respiration, so iron deficiency will cause less hemoglobin production, leading to less oxygen available for energy production. This means the body is short of energy, so the body will be very tired. In extreme cases, it can cause anemia, which is extreme fatigue caused by low iron levels. Girls and women are more susceptible to anemia due to the loss of blood during the menstrual cycle.
Next, structures of large food molecules. Here, you need to know how to describe the structures of the large food molecules, carbohydrates, proteins, and fats. These large molecules are large structures made from smaller basic units, and can be digested back by enzymes. There are two sorts of the food molecule carbohydrate. Firstly, starch. This is made up of the elements carbon, hydrogen, and oxygen. Its basic unit is sugar, and its structure is a chain of sugars. Its digestive enzyme is amylase. Next, glycogen. Again, this is made up of the elements carbon, hydrogen, and oxygen. Its basic unit is sugar, and its structure is a complex branched sugar. Its digestive enzyme is carbohydrase. Now protein. This is made up of the elements carbon, hydrogen, oxygen, and nitrogen. Its basic unit is amino acids, and its structure is a chain of many different amino acids. Its digestive enzyme is the protease pepsin. Lastly, fat. This is made up of the elements carbon, hydrogen, and oxygen. Its basic unit is glycerol and fatty acids, and its structure is a chain of 3 fatty acids joined to one glycerol molecule. Its digestive enzyme is lipase.
Now, let's look at food tests. You need to know how to test for the presence of 4 food groups. These are starch, reducing sugar, protein, and fat. Firstly, the Polysaccharide Test or iodine solution test. Polysaccharides, poly meaning many, and saccharide meaning sugar, are large molecules made from small sugar units. They include starch, glycogen, and cellulose. To test for starch, we do the iodine solution test. An iodine solution is an orange solution. First, put a small amount of food sample on a petri dish or preferably a white background. Then place a few drops of iodine solution on the food sample. Then observe and record the result. When we have a positive result, the color changes from a red-orange to blue-black color. This means starch is present. When we have a negative result, the color stays red-orange. This means starch is not present.
Now, the reducing sugars test, or Benedict's test. Reducing sugars are smaller types of sugars, including most monosaccharides, these are single sugar units, and disaccharides, these being 2 joined sugar units. Examples of these are, reducing monosaccharides are glucose or fructose. Reducing disaccharides are things like lactose or maltose. It must be noted that sucrose is not a reducing sugar. To test for reducing sugars, we do the Benedict's test, using Benedict's solution. This is a light blue color. First, put a small amount of sample in the test tube. Next, add Benedict's solution to the test tube until you have a nice blue color. Then, heat the test tube in a beaker of hot water for 10 minutes. Lastly, observe and record the result. A positive result is when you have a color change from light blue to other colors according to the amount of reducing sugars present in the sample. Green means a low concentration of reducing sugars. Then we have yellow, orange, up to brick red. Brick red means a high concentration of reducing sugars. A negative result is when there is no color change or the change is none of the colors mentioned before. This means there are no reducing sugars present.
Now, the protein test, or Biuret's test. To test for proteins, we do the Biuret's test, using Biuret's reagent. This is also light blue. Firstly, place a small amount of sample in a test tube. Second, add some water. Thirdly, put several drops of Biuret's reagent into the test tube. Lastly, observe and record the result. A positive result is when there is a color change from light blue to a violet purple color. This means protein is present. A negative result is when there is no color change. This means protein is not present. Lastly, the lipid test, or emulsion test. Lipids are the group that includes fats and oils. To test for lipids, we do the emulsion test. Emulsions are small droplets that are insoluble in water. Firstly, if your sample is solid, place a small sample in a mortar. Add a small amount of ethanol and grind it with a pestle. Filter the mixture to collect the filtrate in a test tube. Secondly, if the sample is liquid, add the sample into a test tube. Thirdly, add some ethanol to the test tube containing the sample or filtrate. Fourthly, add some water to the same test tube. Next, cover the top of the tube and shake vigorously. Then, watch to see if the liquid becomes cloudy. Lastly, wait to see if the cloudiness settles. If it does not settle, it is an emulsion. If we have a positive result, there is an emulsion and lipid is present. If we have a negative result, there is no emulsion and lipid is not present. You need to know that if a sample is to be tested for multiple food groups, every test should be done with a new sample. For example, don't conduct the Benedict's test on the solution you just tested with Biuret. Get a new solution.
Lastly for today, the manufacture of yogurt. You need to know, because it is delicious, the use of microorganisms in the manufacture of yogurt from milk. Firstly, milk is first pasteurized to kill all other bacteria originally in the milk. Secondly, milk is cooled to 40 degrees Celsius and then the lactobacillus culture is added. Thirdly, the lactobacillus anaerobically respires using the lactose as the substrate, producing lactic acid. Fourthly, this lactic acid lowers the pH of the milk, denaturing the proteins to coagulate or stick together. Lastly, the yogurt is now cooled and other ingredients may be added. The syllabus says you should be able to, so check if you can, do the following. Define nutrition. List the principal sources of, and describe the importance of carbohydrates, fats and proteins, vitamin C and D only, mineral salts, calcium and iron only, fiber or roughage, and water. Describe the deficiency symptoms for vitamin C and D only, mineral salts, calcium, and iron only. Describe the structure of large molecules made from smaller basic units, like simple sugars to starch and glycogen, amino acids to proteins, fatty acids, and glycerol to fats and oils. List the chemical elements that make up carbohydrates, fats, and proteins. Describe tests for starch, an iodine solution. Reducing sugars, Benedict solution. Protein, Biuret test. Fats, ethanol. Describe the use of microorganisms in the manufacture of yogurt.
Thank you for watching our video. Please like, subscribe and share. And comment below so we can clarify things for you.